Well, I got a little cut off in that video. I only have 15 minutes with each of these videos. That's why they're split into multiple videos. So I apologize. I'm going to go ahead and start at this particular slide just to make sure that you understand exactly what happens in each phase. So this is in an animal cell. This is the whitefish blastula, what we looked at under the microscope last night. In interphase, you're unable to see any chromosomes. You can still see the nucleus, but the chromosomes aren't coiled and condensed. In prophase, you can see the chromosomes begin to coil and condense, and the nuclear envelope begins to dissolve. In metaphase, you can see that they're lined up in the middle. They actually have two metaphase pictures, and they have them lined up in the middle again here. And anaphase, the chromosomes move away from each other. The sister chromatids are separated. And then in telophase and cytokinesis, the nucleus reforms around the chromosomes and the cytoplasm is divided. And this is a cleavage furrow right here that creates in between the two cells. And I'm assuming you can click on each of these. I'm going to go ahead and click on to see where it takes me. Never mind, it's not a good place. One second, sorry about that. Okay, so mitosis in animal cells, it gives you a really good description for each of the phases right here. Prophase, the membrane disappears, centromeres migrate, spindle fibers appear. Chromatin, which is just what they refer to chromosomes as before it's condensed, condenses and chromosomes are now visible, each composed of two sister chromatids, and the centromeres attached to the spindle fibers. Don't forget, interphase is growth and duplication of the DNA. This is actually a really good slide right here. This one has everything all on one page. So if you needed something to study, I went back and I looked at the test that goes with this unit, and there was probably 20 questions that dealt with mitosis alone out of like 60 some multiple choice. And this slide right here is really important that you can at least identify a description of what occurs in each phase. And you definitely need to understand that interphase is the largest time period. You need to understand that the chromosomes duplicate at that point, not double, they just duplicate. So and it's called replication, is when the chromosomes make a copy of themselves where they're attached and they go from a chromatid to sister chromatids. You need to understand that mitosis is prophase, metaphase, anaphase, and telophase. And the specific point of mitosis is to divide the nuclear contents. And it creates two daughter cells, and each of them have a diploid number or a full set of chromosomes. This is just a sum up of exactly the lab last night. Everybody went through and drew each of these phases, and you were supposed to put a description next to each phase, which some of you just described what you saw, which was good, and I took that. But the point is that you can hear a description without the picture and know what phase they're referring to. So they've got quite a bit of a description in here, and this is just from your book. So mitosis in plant cells is just a little bit different than mitosis in animal cells. For instance, this occurs in meristematic tissues, and I thought, they're not going to know what that word meant, so I put a link in here. So what is a meristematic tissue? Basically, they're small cells with thin cell walls. They have large nuclei, very little vacuoles, and not much intercellular space. They're usually at the tip of the roots and the tips of the stems. This is called the apical meristems. It's also in buds and nodes of the stems, in the cambium between the xylem and phloem in dicotyledon trees and shrubs, and under the epidermis of dicotyledon trees and shrubs. So a lot of that stuff's getting pretty technical. Basically, where the plant grows is where you find meristematic tissues. And you don't need to know more than that. I just thought you might want an explanation of exactly what they were referring to. Okay, they have the same phases as animal cells, and plant cells do not have centrioles or asters. Okay, this is the picture from your book. You can see the cell wall and you can see the spindle fibers. They're still not 100% sure about the formation of spindle fibers. That's one of the areas of research, and so this part is confusing to me, but you just need to understand that in animal cells they talk about the centrioles, but they don't talk about centrioles in plant cells. But yet there's still these spindle fibers, and you can still see clear points of connections from the spindle fibers, but there's just no centriole that they refer to. Also, the difference here is instead of a cleavage furrow, they form a cell plate. So the Golgi apparatus makes up all these little vesicles, sends the vesicles to the center, and they form the cell plate, which is the beginning of a new cell wall. So in animals, you need to know this is the cleavage furrow. It forms between two daughter nuclei, and it's like a ring or a string is tied around them, and then it's just pulled until it pinches the cells in two. And so the contractile ring contracts, deepening the furrow until the cells separated. Here's an example of plant cell cytokinesis. It says that you can see the vesicles filled with cell wall components are sent to the center. Remember, the vesicles are just packages sent from the Golgi apparatus, and they appear and the cell plate forms, and then soon a new cell wall's there. And then that's why, you know, that when you see a plant that's in telophase and cytokinesis, it takes up the same space as a cell that's in interphase. It's because after it comes out of cytokinesis, it enters back into interphase and it starts to grow. And so it has to grow before it can be duplicated again. Here's the phases all in order with the diagram just to help you study again. Notice in interphase, you can see that they've already duplicated the centrosomes. The chromosomes can't see, they're called chromatin at this point. In prophase, the chromosomes start to condense, the centrioles migrate to opposite ends of the poles, the nuclear envelope disintegrates. Metaphase, the chromosomes line up so that their sister chromatids can be separated and migrate to opposite ends of the poles. So in the middle, the chromosomes are lined up in metaphase, and then in anaphase, the chromatids are now called chromosomes, and they can be migrated to the opposite ends. And so anaphase, they move away. The sister chromatids are broken apart to form chromatids, and they move away. And the telophase, the nuclear envelope begins to reform around the new chromosomes, and the cell begins to pinch into two. In cytokinesis, that's whenever it's pinched into two new complete cells. Here's one more picture from the onion root tip, and they have the phases nice and labeled for you. You can see all those little arrows are pointing to different cells that are in interphase. You can see in the second box, prophase, you can see the chromosomes are starting to coil up and become visible. In the third box, prophase, again, the chromosomes are beginning to darken and be able to be visible. Metaphase in the fourth box, lining up in the middle, anaphase moving away, and in the last box you can see there's that one little tiny cell that has two obvious nucleuses and the cell plate forming, and notice that it's almost the same size as this cell up here that's in interphase. Here's another one. You can see this one is probably a pretty young cell because both of those cells are still fairly small. They still have to go through the growth. Like this one has already gone through all of its growth. You can tell it's probably going to start dividing soon. Here's a nice anaphase down here also. And that's pretty much it. I'm going to post some questions that you need to be prepared to answer, and I'll give you a multiple choice quiz on Tuesday over everything that's been in these videos and everything in your chapter. And I'm not going to touch the meiosis part. You need to be go ahead and print that slideshow and bring it to class with you. We'll do it in the next class together. And so basically you're going to need to know up to page 88. That's going to be where the quiz questions come from. So have a great weekend. I hope this was okay. Give me some feedback so I'll know if weather's bad again, this is a way we can do it, or if this is just not acceptable. Just give me some feedback, and I'll talk to you later. Goodbye.